Good morning, afternoon, and evening. It's Thursday, April 9th, and on behalf of everyone at Simulations Plus, welcome to day 2 live stream learning event. As the globe continues to grapple with increasing challenges to sustaining business as usual, we hope that being able to provide our scientific talks and scheduled poster presentations for the now canceled SOT Tox Expo and Annual Society of Clinical Pharmacology and Therapeutics in a digital format is a valuable experience for you. For optimal sound and audio connection, we highly recommend you close any additional web or streaming applications that may affect your internet bandwidth. Presenting today will be John DiBella, James Mullen, Michael Lawless, and James Clary with the GPX and APX demo at the end. So get comfortable, it's going to be a busy 90 minutes. An opportunity to ask questions will follow John's talk and after each poster session. You may send your written question using the questions pane on your control panel, or you may use the hand-raising icon on your control panel to ask your question directly. Please be sure to enter in the unique audio pin displayed when you join the call. This call is being recorded for playback on www.simulations-plus.com. Now, before we get started, we have a quick poll question. Do you use modeling and simulation in your current research activities? We'll give you about 30 seconds to vote. Go ahead and submit your answers now. A few more seconds left, and it is overwhelming. We're going to close the polls now. And it looks like 94% of you are currently using modeling and simulation, so you are in the right place today. Now it is my pleasure to introduce Mr. John DiBella. John leads the Simulations Plus Lancaster division, overseeing the development of ADMET Predictor, GastroPlus, PK Plus software, and consulting projects. In addition to traveling the globe, hosting training seminars, and presenting at many scientific conferences. John, take it away.
Thanks, Shakul Kat, and thank you, friends of Simulations Plus, for carving out time today from your busy schedules to join us. We hope you're all staying safe, healthy, and at home. And what I would like to do over the next 20 minutes or so is give an update on our efforts to marry various technologies to support next generation risk assessment with a focus on chemicals. We do many presentations talking about how our technologies can be used to drive and guide pharmaceutical research. And today, we want to give a tip of the hat to our users in the chemicals, consumer goods, and non-pharmaceutical markets. Before I start, please allow me to spend a couple of minutes discussing a few updates at our company. As many of you know, Simulations Plus is a global organization with over 120 employees, devoted exclusively to the development and application of modeling and simulation solutions. This graphic that you see on your screen outlines the various tools we develop and where they typically are applied in the drug development process. We are proud to mention and note that a significant portion of our revenue is reinvested into software R&D each and every year, as we make it a major goal and priority to continue to innovate and provide new functionality that you, our clients, request. Also note that we offer consulting support on a project-by-project basis, and as we have entered into this strange and challenging time today, we are being contacted by many companies who, with experiments and trials paused, wish to generate unique insights with the data on hand by using our technology and engaging with our experts. Now, we've had lots of exciting announcements over the past 12 months, including entering into numerous funded collaborations that have started with the FDA, large pharma and biotech companies to implement and, more importantly, validate new functionality. Just last week, however, we released news that will truly lift us to another level, the acquisition of Lixoft. Many of you are familiar with the company and their tool Monolix, which is an easy-to-use software platform to build and evaluate population PK/PD and systems pharmacology models. I just encourage you to stay tuned for updates on how we will be integrating Monolix with GastroPlus, Kiwi, the DiliSim platforms, and all of our other tools. Really, the sky's the limit in terms of where we're going to be going as a company, providing you with solutions over the next 12 to 24 months. And finally, as a company, we have adjusted our standard business priorities and schedule to provide immediate support for pharmaceutical research and development efforts to address the COVID-19 virus. This crisis has, in our opinion, crystallized an improved discovery paradigm that will facilitate a speedy rebound for the industry and may finally make real that long sought goal of lower cost, faster to patient, and safer drugs. We stand ready to help in any way we can, and we do encourage you to reach out through the dedicated email address you see on your screen. So thank you for giving me those few minutes there to do the introductions. Now what I'd like to do is just talk about a few statistics and then set the stage for the meat of the presentation, focusing on risk assessment for chemicals. As we've grown our user base over the years, we've always been able to point to how our software and services can help companies make very important internal R&D decisions. And what we've seen significantly grow over the last several years especially is how the results from our tools are being applied to inform regulatory interactions. I'm very happy to say that what you see here first, this number, over 35 approved drug products that are on the market today, were supported by GastroPlus simulations. These simulations were reviewed and accepted by global health authorities. And this number is a floor. We've just surveyed a few of our customers to get some feedback on how they've applied their results to support regulatory interactions, and we're pretty confident that this number is quite a bit higher. And these applications are across a variety of different areas. There are on the market today products which were supported for metabolism-based interactions, transporter-based interactions, establishing specifications around critical material attributes or products, and in some cases waiving BABE studies. So as you're starting to think about how we can apply these tools for internal purposes, don't lose sight of the long-term goal of how these results will ultimately help you inform your interactions with health authorities. The second number I'd like to leave you with today, the user base for our technology, over 250 companies and regulatory agencies licensing the platform today. That's a huge number, and it reflects not only this growing adoption of modeling and simulation, but more specifically, the comfort level that users are finding with our integrated set of tools. And where we're seeing significant growth over the last several years is on the use of the technology for non-pharmaceuticals. Approximately 20% of this number here are companies in chemicals, consumer goods, cosmetics, areas that 10 years ago we hadn't really even considered. And I'm going to talk a little bit more about how it is that we've been able to make such advancements in these new markets with some of the enhancements to the underlying software tools. And finally, over 1,000 peer-reviewed journal publications and conference presentations have been published, have been given at various conferences globally, citing results from our software tools. So when you are getting ready to engage at the regulatory level and you're getting questions back from some health authorities about the platform that you've used and whether it is qualified for certain purposes, all we have to do is point to this long, long list of publications and conference presentations. And the majority of these are not coming from Simulations Plus. We make our best efforts to continue validating and verifying software results with data that we find from literature or get access to from our collaboration partners. But the majority of these publications and presentations are delivered by our users. And we've got a number of publications on a diverse set of compounds, a diverse set of applications and purposes, making this, again, one of the most qualified software programs and platforms in the world. Now, one such example, as we started to build up confidence over the years in terms of how our tools are going to be used for first pharmaceutical research, came from work like this at Pfizer, where now over 10 years ago, researchers wanted to evaluate how classical methods, allometric scaling, compared to, at the time, a newer approach, physiologically based pharmacokinetics to predict first-in-human exposure following oral or IV dosing. And so in this work presented initially in 2008 and then followed up with publications in 2010 and 11, Pfizer evaluated for over 20 of their clinical drug candidates how well they would be able to predict their phase 1 trial results with in vitro inputs only using PBPK or taking some of their preclinical PK data, scaling that information to a human environment and generating predictions in that manner. And so these are some screenshots from the posters and publications outlining the set of inputs, a classical set of ADME properties, physicochemical parameters that would go into the models versus applying PK data generated in rats or dogs, calculating the non-compartmental clearance or volume of distribution, and scaling or translating up to humans. And very proud to say, when these results were presented and published, that the GastroPlus platform was ranked #1 in predicting everything, following IV dosing, following oral dosing, predicting volume of distribution, clearance, AUC or Cmax. And this is compared to other PBPK software, and more importantly, compared to the allometric scaling routines that were commonly performed at Pfizer. So this is just one example of publication showing how results from GastroPlus have been validated and or verified against real PK data. Now, when we're talking about these validated system models in GastroPlus, for us, creating this virtual human or animal environment is going to consist of two components. First, we would have the physiologically based biopharmaceutics model or mechanistic absorption model, which describes our site of administration. And don't get tricked by the product name GastroPlus. The program does have these PBBM models for all major dosing sites around the body. as you see in that figure on the left. And all of these models have been developed through funded collaborations with industry and or the US FDA. So we're using these detailed multi-compartment descriptions of the site of administration to track local exposure. And we're going to connect then the second component to the model, which is going to be the PBPK system, to then simulate and predict concentrations throughout the body. And in GastroPlus, we provide validated systemic PBPK models for human and 6 preclinical species. And for humans, we have these validated models in place for a variety of different population types, health status, and age groups. Now, as we have spent a lot of time over the years validating these system models, validating the underlying algorithms and equations for predicting pharmaceutical drug exposure, we've now made a bigger investment to try and see how our tools can be applied towards chemicals. For assessing risk, safety risk, especially for chemicals, the traditional approaches have always been to try and perform animal testing across various exposure levels to measure what we would call an adverse organism response. And from that, then extrapolate to humans to try and estimate the safe dose. These traditional approaches have a slow throughput and required extensive animal testing. And as many of you know who work in this space, there's been a real push to try and eliminate animal testing and find alternatives for assessing risk. Toxicity testing now in the 21st century is going to start to incorporate more mechanistic modeling approaches and trying to find the best ways to leverage our in vitro data to extrapolate to an in vivo environment. And what we're going to try and do is utilize and apply a panel of pathway-based assays, which could be cell lines, organelles, organs on a chip. Use some of that system biology-based toxicity information and incorporate that into PBPK, or for chemicals, PBK modeling to estimate human exposure. Here, there's real potential to do this in a high throughput manner, as quickly as we can generate a lot of that in vitro data, and more importantly, no animal testing, meeting the goals that are being established globally. Now, what are we trying to do to address toxicity testing in the 21st century? Next generation risk assessment for us means saying I do to the marriage between different modeling technologies, machine learning, PBPK, and quantitative systems toxicology or pharmacology models. The idea being that we could try and take chemical structures like we see on the left, apply machine learning models to predict a variety of different ADME-tox endpoints that then seamlessly fill in the, help us describe our chemical properties within a virtual animal or human system. And we can start to predict, following different administration routes and parameters, what would be the local and systemic exposure, the distribution throughout the body, if we have any metabolites that are being tracked, predict both parent chemical and metabolite levels, and also patient variability. And we can do the first two today, machine learning and PBPK, and I'll talk about this coming up in some examples. And our goal over the next 12 to 24 months is to see if we can't further integrate some of the QST, QSP models from our DILIsym services division to then be able to predict more precisely the impact of changes within a specific tissue, like the liver, the kidneys, the lung, or the heart. A very important publication validating this marriage between machine learning and PBPK was published in 2018 from Dow Chemical. Dow Chemical performed an evaluation across several dosing routes, utilizing a mix of chemicals and pharmaceutical drugs, their structures only, to predict what would be the systemic PK exposure following oral, dermal, or inhaled administration. And for toxicokinetic purposes, we can predict within tenfold, according to the chemicals evaluated by Dow Chemical, over 80% of the compounds from the dermal and oral routes and over 60% from the inhaled route. There was also some additional validation which was performed comparing the predictions of different endpoints from our machine learning models versus competitor tools as well. And I just want to kind of highlight here, because we often get the question, your underlying databases, training sets for the machine learning models most likely comprise a majority of pharmaceutical drugs, or most of those over 1,000 publications are likely focused and referenced on pharmaceutical drugs. And chemicals can often have different clearance mechanisms, uptake mechanisms, and so on. And I just hear from that publication from Dow Chemical, they list the compounds that were evaluated. And if you take a closer look at the publication, you'll see that the majority of compounds evaluated were chemicals, not pharmaceuticals. And to be able to have the level of acceptable accuracy needed for toxicokinetic purposes validates and verifies or qualifies the GastroPlus platform for chemicals, and more importantly, this machine learning PBPK marriage. Now, for the remaining few minutes here, what I want to do is focus on recent improvements that we have made to the dermal PBPK model in GastroPlus. So while we have validated PBPK models for all major dosing or exposure routes around the body, through this coordinated production today, you're going to hear more in a few minutes on what we've been doing with our oral and inhaled models to guide risk assessment for chemicals. So there's lots of good stuff coming up. Now what I want to do is just spend a few minutes discussing dermal. So as we've mentioned, we've been very lucky to have partnered with great companies, great organizations on the development of these exposure models. And the story with our dermal PBPK system starts in 2012 with GlaxoSmithKline. We initially developed and validated with them an in vivo model over a 2-year period. And you can see that model structure on the right. We have compartments that describe the vehicle, which is going to be typically applied on the top of the skin, and all major skin layers, with models in place to estimate the relevant diffusivity, partitioning, absorption to track both local and systemic concentrations. The default physiology models can be seen in the middle of the slide, and you have the option to build your own systems without the need to write any equations. A follow-up project with GSK included the modification of the in vivo model to also simulate and predict concentrations in an in vitro system, which we see on the left. And here what we're trying to do is specify parameters in the donor compartment and simulate how much appears in the receiver compartment. Our long-term goal is to utilize measured in vitro data to parameterize the permeation through the various skin layers and extrapolate that information to better predict in vivo exposure. Truly an IVIVE for dermal. In work presented at the 2018 Society of Toxicology meeting, Cosmetics Europe presented the results from their evaluation of 6 in silico skin penetration models. The consortium rigorously evaluated GastroPlus and several other commercial and academic dermal models on a number of criteria. And we can see some of these results here, including prediction accuracy using blinded data for 25 diverse compounds or chemicals. And the results had GastroPlus ranked #1 compared to other PBPK software, other academic models which were developed. Now, from that work, we entered a funded collaboration project with Cosmetics Europe to add new functionality to the GastroPlus TCAT models, both in vitro and in vivo, to support the push for alternatives to animal testing for risk assessment. In this partnership, we have introduced new approaches to predict chemical evaporation kinetics from the top of the skin, improved methods to predict permeation through the skin layers, and integrated saturable metabolism kinetics within the skin to be able to predict and track local exposure for both parent compound and any metabolites. Not only have we enhanced the underlying GastroPlus models, but we've also been very fortunate to validate these improvements with data provided by the consortium members, data that no other company has access to. We've also been very lucky to receive a second grant award to improve our dermal models, this time from the US FDA. And the focus of this project is to incorporate more formulation and administration properties and attributes into the models, with the goal to build up confidence in the predictions to, in the future, better assess local bioequivalence for different products. There are also tasks, however ambitious, to develop in vitro models to better evaluate the impact of excipients on skin permeation for active compounds. So for example, we have added heat-separated epidermis and dermatomed skin assay models to the program. A mini consortium consisting of the FDA, pharmaceutical and generic drug companies, and CROs are contributing expertise and, equally or more importantly, data to help with model development and validation efforts. So there's lots of good stuff going on in the dermal modeling space, and we are now starting to see the fruits of our labor. A new publication from Unilever Safety and Environmental Assurance Center Group was published just a couple months ago, outlining the application of physiologically based kinetic modeling for chemicals to assure safety of consumer products without animal testing. In this publication, Unilever proposed a really novel framework based on the construction of these PBK models in GastroPlus, describing consumer habits and practices, formulation types, and ADME properties for new ingredients in dermally applied products. Building off on initial in silico estimates for inputs into these PBK models, sensitivity analysis is performed to identify critical parameters and guide in vitro experimental designs. And the framework that they published is that diagram that we see here on the right. In this next generation risk assessment decision tree, levels 0 and 1 serve to help identify the use scenario and define the inputs using a combination of in silico and or in vitro methods. So the focus of this particular work was on evaluating the impact of different use scenarios on coumarin. And so Unilever first identified 4 common use scenarios as defined by the European Consumer Exposure Studies, and easily set them up for simulation in the GastroPlus TCAT model. And this is what we see in this table here in the upper right. So for face creams, body lotions, shampoos, or kitchen cleaner liquids, we have some estimates on what would be the amounts of the product used per day, frequency of use, application site and skin surface area covered, and so on. And we can define these use scenarios for these different products within the GastroPlus model. They then ran the coumarin structure through the ADMET predictor machine learning models to generate the set of ADME inputs for the PBPK simulations and compiled available in vitro data for comparison. And that's what we see in this bottom table here. Very common set of ADME inputs, similar to what we saw from that earlier Pfizer work, which was focused on validating PBPK models for pharmaceuticals. Same type of inputs. We do have some additional models here to be able to predict some of those partition coefficients and diffusivities from chemical structure. But what's also interesting here, and we'll see on the next slide, is how Unilever was able to apply some of their in vitro skin penetration data and use that to help calibrate some of these parameters describing permeation through the skin. When we get to that level 2 exploration, where we want to start doing the in vitro and vivo extrapolations, what we're going to try and do is leverage that in vitro data to predict in vivo exposure, and if possible, validate versus any available PK information. Measured PK data, we know, is becoming rarer to generate, and so the $64,000 question to answer is, do in vitro points of departure, and we see that here in this section of the decision tree, help us confidently derive margins of safety. In Unilever's publication, the in vitro data that was generated for coumarin from the Dermatomed skin assay was used to calibrate the permeation parameters, like we saw in that previous slide, through the various skin layers using that in vitro TCAT model in GastroPlus. And this is what we see, if I blow up this diagram here, some of the comparisons between the simulated and observed concentrations in the receiver compartments following different administrations of coumarin, different vehicles. So using that data to help us define permeation through the skin, we then apply those values to our in vivo model to predict systemic exposure. And what we see in this plot, this diagram in the lower right of the slide, is the distribution of systemic Cmax values from the population simulator in GastroPlus following administration through different topical products using either in silico or in vitro clearance values as the input. So we're able to apply some variability, generate a set of virtual subjects in a population group, and help us establish our margins of safety for risk assessment purposes. Now, where do we go from here? With modeling technologies becoming more and more advanced, we're really starving for data that can be used appropriately to help predict new mechanisms reliably from structure. So for example, transporter interactions, phase 2 metabolism kinetics, and more. Integrating these predictions into models that allow us to simulate exposure across multiple routes at once. For example, a chemical that is ingested, inhaled, stuck on the skin, and mimic real world outcomes for chemicals with confidence is also going to be important. That being said, while we still have some work to do, we can use alternatives to animal testing today, especially through the use of mechanistic modeling and simulation, and with a call out to health agencies responsible for regulating chemical registrations. Remember that proprietary modeling platforms are not to be feared, but embraced for the reasons that we see on this slide here. Let's try to accelerate the use of mechanistic modeling and simulation through platforms that are being utilized on a daily basis internally at companies to help support chemical registrations in a more efficient manner. Let's continue to eliminate animal testing and produce safer chemicals for ourselves and for Mother Earth. So thank you for your time today, and please stay safe, stay healthy, and most importantly, stay home. Arlene?
Thanks, John. That was insightful. A reminder, an opportunity to ask questions. You can do this by sending your written question using the questions pane on your control panel, or you can use the hand raising icon on the control panel to ask your question directly. It looks like our first question coming in from Joe. John, is it possible in GastroPlus to simulate multiple chemical exposure routes in a single model? For example, he asks, chemicals which may be ingested orally, inhaled, and exposed to the skin at the same time or even different times?
Yeah, that's a very good question, and it's something that I called out on the future direction slide. Fortunately, that will become a reality in the next release of GastroPlus, the next generation platform called GPX. We will have opportunities to be able to set up simulations where we do dose a certain amount at a certain application or skin surface area, while also inhaling a certain amount of chemical through the nose or through the mouth, while also ingesting a certain amount of that chemical to see what the GI absorption looks like. So trying to mimic that real world outcome is something that will become a reality for us with the GPX platform.
Great. Well, we should stay tuned then for the end of our 90 minutes or a little under an hour now for that GPX demo. Now we're going to change pace and we are going to do a poster presentation. The first poster is presented by James Mullen, Senior Principal Scientist. James has over 15 years of experience in computational modeling in the pharmaceutical industry, spending 10 years at Ben Research before joining Simulations Plus in 2014.
Hello, everybody. This is James Mullen, and I'm here to present to you a generalized PBPK model for evaluation of inhalation exposure of volatile organic compounds. I am a principal scientist at Simulations Plus in the Hollywood division here in Lancaster, California. I work in the simulations technology group, and I'm responsible for helping on the development of GastroPlus, Membrane Plus, and DDD Plus software here. I'd like to talk to you a little bit about our physiologically based pharmacokinetic model that we built for inhalation, and we believe it's a valuable tool to evaluate the inhalation exposure of volatile organic compounds for toxicological purposes. Many literature models are sort of ad hoc and built for a single purpose or molecule, and generally include a small subset of tissues and rely on experimental or fitted partition coefficients. Of course, in GastroPlus, we have a standardized PBPK platform that allows you to predict partition coefficients based on a number of different types of models, like the Lukasheva method, or the Rogers and Roland method. And you can utilize that to predict the vapor inhalation using our pulmonary model. Previously, our model was built on sort of dry powder, nebulized, or solution formulations, but we've added this additional capability to our GastroPlus 9.7 platform, and it'll be available in subsequent releases of GastroPlus. So in terms of the model that we've built, it was built in GastroPlus version 9.7, mass balance equations that describe the gas flow into and out of the system were developed to calculate the VOC in the different lung sections. So if you look on figure 1, you can see our model schematic for our pulmonary model in GastroPlus, where we've kind of segmented the lung into an extrathoracic, a thoracic, a bronchial, and an alveolar region. So previously, we didn't have a vapor compartment in these different sections, but as you can see in figure 2, we've added that vapor compartment to the different sections of the lung. And then the gas flows and the concentration of vapor is going to be tracked based on the flow rates and the inlet concentrations of the system. The calculation of the partitioning between the gas phase and the mucus of the lung is determined by the Henry's law constant. And so we have, there's a lot of experimental data for VOCs that's available from the EPA, and also we have an ADMET predictor model that can predict the Henry's law constant. So with those additional equations and physicochemical properties that were calculated from ADMET predictor 9.5, we could calculate the vapor exposure for some volatile organic compounds. We can also predict the partition coefficients in the PBPK model using the Lukachova method. And then based on some experimental data, we can define the metabolic clearances of the compounds, test compounds, and see how well we can fit in vivo data for inhalation exposure in rat and human. So the validation cases that we looked at here are for ethanol, methanol, acetone, and styrene. Of course, there's a ton of data on ethanol because of its usage in roadside tests for drunken driving in this case. So there's a lot of data for ethanol. There is also a lot of data for, industrial exposure for methanol, acetone, and styrene. So first we'll look at methanol inhalation in rat and see how the predictions in rat can be then scaled up to human. So in figure 3, I'm showing the predictions for methanol inhalation in rat from 200 ppm dose on the left-hand side all the way up to 2000 ppm dose on the right-hand side. So we're able to do a pretty good prediction at both 200 and 1200 ppm in rat. At the highest dose, we might be under predicting slightly, but it looks like the 2000 ppm dose is greater than dose proportional. So maybe there's some other metabolic processes going on, maybe some saturation or whatnot. We use the exact same PBPK settings to predict human. So the same partition coefficient model, the same model setup. The only difference we did was change the physiologies from rat to human. We also adjusted the clearance of methanol in human. There was one data set where we had the sort of intravenous clearance of methanol after an incidental exposure that was measured in the hospital. So we were able to use that to calculate a KM and Vmax value for specific to human. And then we used our vapor inhalation model to predict either a 0.5 hour inhalation of 800 ppm, a 1 hour inhalation of 800 ppm, or a 2 hour inhalation of 800 ppm in human. And those results are shown in figure 4. You could see there's a pretty good fit to all 3 of those cases. So we're able to fairly well reproduce the dose sort of dependence in methanol based on the inhalation time in human. The error for the average Cmax and AUC for the human model was 13.4% and 19.9%. On the next slide here, we'll be looking at predictions for ethanol, acetone, and styrene. So In figure 5 are our predictions in human for ethanol. So from 126 ppm all the way up to 1050 ppm, we were able to predict the dose dependence for 4 hours of exposure of ethanol very well. We also, on figure 6, predicted the inhalation of acetone in human very well. In this case, we were able to train the clearance in the model through some IV data of acetone in human subjects. And then the final model we have down there in figure 7 is for styrene. So for 50 ppm, 64.5 ppm, and 80 ppm, we've done a fairly good job of predicting the exposure of styrene. It looks like one of the data sets is abnormally low concentration is predicted for 80 ppm, and it doesn't quite correspond to some of the other literature data, so it might just be an issue with that literature data set. Is why we're not predicting as well for the 80 ppm. So overall, in the 4 compounds we've looked at here in this case, we were successful in predicting the plasma concentrations for rat and human, and showing that the model can really be used to scale from an animal species. Once you have a model built in rat, you can really apply the same settings to a human subject and predict a good result for your exposure. So with that, I'd like to thank you for watching, and you can find my poster and others like it in our resource center. It's www.simulations-plus.com. And I look forward to answering your questions on this model here. And thanks for letting me present today and show you the new work we've performed on the pulmonary inhalation. Thank you.
Thank you, Jim. Okay, so a couple of questions are coming in now. We've got a question here. Although we're talking about organic volatile compounds, do you think that this simulation models could be applied to API foam generated in R&D labs or manufacturer sites?
API, what was that, foam?
Yes.
Or foam. I mean, it's not a vapor, so I mean, it would have to be vaporized, right? If it's some sort of a foam is a liquid that has, you know, air sort of, I guess, pumped into it. So, I mean, I would probably use the solution model for our pulmonary model, probably to simulate something like that. This is specifically for vapors. So, if there was an API that was vaporizable, then sure. Or the other thing it can be used for, if any API is volatile to any extent, if you take it intravenously, this model will also predict how much drug you'll expel via exhalation clearance. So basically it will work for any drug that is in systemic circulation, and it's volatile enough to basically exhale in your breath. So you know, that's one of the mechanisms of clearance of ethanol from your body is you breathe some of it out, maybe like 1 to 2% of a dose, a liquid dose of ethanol you exhale in your breath. So any API that could apply to as well, but if it's in sort of a liquid foam state, I would probably guess it would be more like a solution of a liquid and wouldn't sort of apply to the vapor modeling.
Thank you, Thalita, for that question. Moving next to Andy. Are the simulations fitted separately for each data set or together?
No, so these predictions all were the systemic clearances all fit to IV data in the cases of the I think all of these cases here. So really the clearance was defined by an intravenous dose, and the PBPK model settings were all basically built on IV and oral doses, and then they were applied to the pulmonary model. So in all these cases, there's really not any fitting going on other than fitting against oral and IV data to get the proper parameters to then apply to the pulmonary model. So really there wasn't, a lot of fitting going on. The one case there was some fitting was where there is some lung metabolism going on, in which case you would need to do some level of fitting. So in the case of, I think styrene, we had some lung metabolism included in the model, but for the most part, yeah, these aren't fitted across the data sets.
Okay, great. And for the final question, I'm going to put Hiro on the line live to ask his question. So here we go. I'm going to unmute you, Hiro, for your question. You're live now with Jim. Go ahead. All right, maybe he's having some technical difficulties, so we will catch up with his question after a short poll question. Which modeling technology are you guys using in your research activities? You've got 30 seconds, and if you could submit your answers now. Just a few more seconds left. PBPK is closing in strong, and we're going to close the poll now. It looks like PBPK is the definite winner at 73%. So all of you guys continue to listen. You are in the right place. We're going to go right into the next presenter, which is Michael Lawless. Michael is Senior Principal Scientist for Cheminformatics Solutions at Simulations Plus. Dr. Lawless is a computation chemist with expertise in QSAR modeling to predict admit properties. He also hosts many of our online tutorial videos for Admit Predictor and GastroPlus.
Hello, I'm Mike Lawless, Senior Principal Scientist in the Cheminformatics Group at Simulations Plus. The title of my presentation is Using in silico in vitro to in vivo extrapolation to predict the oral dose required to activate the aryl hydrocarbon receptor. Activation of ARH can have toxic effects on mammalian host. The compound needs to achieve an in vivo unbound plasma concentration higher than its AC50 to produce toxicity. We used in silico and in vivo parameters to perform toxicokinetic simulations to determine the dose required to reach a total plasma concentration equal to the unbound AC50 value. AC50 values were extracted from a Tox21 assay downloaded from PubChem. In vitro, fraction unbound in plasma and hepatocyte clearance data were extracted from the HCTK R package. And this was just used as reference data from the literature, essentially. Then all other input parameters, such as solubility, log P, pKa, were predicted from machine learning models in Admit Predictor. The final data set consisted of 160 compounds that contained AC50, FUP, and intrinsic clearance data. The data contained agrochemicals, pharmaceuticals, and industrial chemicals. This next slide illustrates the overall method. The ACAT model is used to simulate immediate release tablet going down into an oral dose of an immediate release tablet, and it uses the ACAT model, which is has 9 compartments for the stomach, starting in the stomach where the compound would dissolve, and then it would work its way down the GI tract and be absorbed into the gut wall. Some of the important parameters here that we'll look at and discuss are solubility and permeability. Once the compound enters the enterocytes in the gut wall, this is the fraction that is absorbed for the molecule. The molecule then transits into the portal vein where it can be metabolized into the liver. And this is where we'll use the hepatic clearance and the FUP in vitro values to look at metabolism in the liver. On the other side of the liver, we term this the oral bioavailability of the compound. Then it will enter systemic circulation where it can be absorbed into the tissues. It can be cleared renally. Here we're using, again, the fraction unbound to plasma times the glomerular filtration rate for the kidney clearance. Then it can also go back into the liver and be metabolized and cleared there. In the end, what we're doing is producing a CP time curve. And the program starts with a 10 milligram dose and then uses an analytical method to approximate the time to reach steady state. It then performs a simulation, and if the steady state concentration here shown in this last value here is above the, if the average between the peak and the trough is above the target concentration, it would terminate. If not, it will either increase or decrease the dose until it reaches the target concentration. After 1,000 milligram dose, it basically gives up. So that's our upper level of the dose. These three plots on the left show the distribution of the ARH potency in micromolar quantities, the HTTK fraction unbound to plasma, and the log of the human hepatocyte clearance values. The AC50s range from 43 nanomolar to 74.1 micromolar. The FUP values range from 0 to 1, and 38 compounds have FUP values less than or equal to 0.05 or 0.005. So those are really highly bound compounds, highly plasma protein bound. Anything below 0.005 was estimated as 0.005 in the simulations. The AC50 values were then divided by the FUP to give the unbound AC50 value. So that's the target concentration or the total plasma concentration as input into the program. This graph here in the middle shows the dose, or that shows the results. So this is the estimated dose versus the log of the total plasma concentration. And 18 compounds had a predicted dose of under 1,000 milligrams. The rest of the compounds are shown up here as kind of these vertical, or excuse me, horizontal line points here for them. So this molecule down here is the one that's predicted to have the lowest dose. It is isoniazid, shown here in this plot, and this is the CP time curve for this compound. So the activation, the AC50 value for this is 0.573 micromolar. It's fairly unbound to plasma, so 90% of it's free, and then it has a pretty low human hepatocyte clearance of 1.1 microliter per minute per million cells. So when you take the AC50 divided by the fraction unbound, you get the unbound value. So this is the target plasma concentration, and we're predicting that a dose of 13.4 milligrams would reach this plasma concentration. And so here we have the CP time curve for this dose, and you see that it goes up above the target concentration, which is here in red for that. So that's the kind of the most active compound. Now, a number of these compounds that couldn't achieve the dose below 1,000 milligram had very low solubility. So what we're doing here is plotting the fraction absorbed versus solubility. And for very low soluble compounds, these have a very low fraction absorbed, so they're not can't even enter into the portal vein, let alone get systemic concentrations. And so this CP time curve here is for an example of a compound that had very low solubility. So it's this 3-ring system with the carbonyls here, going to make it a very flat compound, and it has a very low predicted solubility of 0.9 micrograms per milliliter. The AC50 is 3.2 micromolar. Now, that actually is a fairly high value. If you think of pharmaceuticals, typically at their target, they're going to have a single digit or low nanomolar IC50 or EC50 values. So 3.2 micromolar, would be pretty high. The fraction unbound to plasma is 3, so very low fraction unbound. The clearance is a little bit higher, 30.8 microliter per minute per million cells. And then dividing the AC50 by the fraction unbound means it has to get up to 100 micromolar concentration. And it was not able to achieve that even at a dose of 1,000. It does reach steady state fairly quickly here, but it's nowhere near. You see the Cmax is 0.9 micromolar, which is 100-fold short of the target concentration. And then on the right, I just show, instead of a low soluble compound, I show a low permeable compound. And that's this red dot here on the right here. So at its dose, the fraction absorbed was a little over 50%. This compound has a fairly high AC50 of 61 micromolar, medium fraction unbound, very low or 0 for the clearance. Okay, and this was probably, this is an in vitro value, fairly high unbound plasma concentration. The fraction absorbed never got above 54%, so that limits its oral bioavailability, even though it's not being cleared very much in the liver on first pass metabolism. The Cmax gets up to 26. You can see it reaches steady state here at at this time period in here. But the Cmax is only 25, and again, it's much less than the target concentration here. This final slide just shows a summary. So the AC50s were divided by the FUP in order to get the total plasma concentration needed to activate ARH. Compounds with that were very highly protein bound needed to reach a target concentration 200 times greater than their AC50 value. 18 compounds were able to reach their unbound AC50 value with a daily dose of less than 1,000 milligrams. These compounds typically had lower unbound AC50 values, so the average for these was 70 micromolar compared to 1,715 for the other 140 compounds. 17 out of 18 of these compounds had very high fraction absorbed, above 90%. The compounds also had low human hepatocyte clearance with an average of 1.2 microliter per minute per million cells, compared to 51 for the ones that were not able to reach their target concentration with a 1,000 milligram dose. Thank you for watching. You can find my poster and others like it in our resource center at www.simulations-plus.com. I look forward to answering any questions now.
Thank you, Michael. Okay, so the question that came in rather quickly was, I'm currently using ADMET Predictor 9.5. How can I make repeat dose CP time curves like the ones in your poster?
Okay, thank you for the question. So the poster, I ran the simulations in ADMET Predictor 9.5, but then to use the plots, I used a beta version of the next version of ADMET Predictor, which will allow you to do multiple doses and produce a CP time curves of multiple repeat doses day after day. So in the next version of ADMET Predictor, we've enhanced the HTPK simulation module. And when you perform a dose estimation, it's not only going to give you the dose, but it will also give you the time needed to reach steady state, or the number of days, for example, to reach steady state. And then when you create a CP time curve with the HTPK module, there'll be a new input on the dialog box, which includes the number of repeat doses. And so that will be available in ADMET Predictor X, which we expect to release sometime in the summer.
Okay, and now has this model accounted enterohepatic cycle?
No, it's a simple or more simple simulation than that. So In the simulation, we only consider passive absorption. We don't consider gut clearance, and we don't consider enterohepatic circulation of the compound. But that is available in GastroPlus. So the idea would be to maybe do your initial predictions in ADMET Predictor, and then as you increase the complexity of the model, you can run that in GastroPlus, which can account for enterohepatic circulation.
Great, thank you, Michael. Okay, before we move on to Dr. James Clary's poster, I wanted to remind everyone that if you are having some audio issues with the poster presentation session, you may adjust your computer speaker settings. That's where the audio is coming out for these poster presentations. So if you need to adjust a little, we'll give you a couple of seconds here. And let me introduce our final poster session, hosted by James Clary, pharmacometrician for Cognigen Corporation, a Simulations Plus company. Dr. Clary has participated in oncology research through modeling drug concentrations and effect, receptor occupancy and tumor growth of discovery compounds in mice.
Hello, my name is James Clary. I'm a pharmacometrician at Cognigen Corporation, and I work in the pharmacometrics department. And today I'll be talking about optimization of malaria volunteer infection study designs using clinical trial simulation. So a brief history of malaria trials is, with a traditional trial, we would have 3 consecutive cohorts with 8 patients apiece at a low, mid, and high level dose. The problem with this is that if the doses don't adequately establish the dose response relationship, then there is no ability to correct within the trial, and you have to wait really until the end of the trial to get those results. So previous work had proposed a adapted design where we have a single cohort of 8 patients, 2 at the low level, 2 at the middle level, and 4 at the high level of dosing, and then using modeling and simulation to determine if the dose response relationship has been reasonably established. If it had, we can continue on to phase 2 studies, and if not, it gives us a chance to create an additional cohort with different doses in order to define the dose relationship, the response relationship. So the way that we approached this was to develop a platform to assess trial designs. Using stochastic simulation, we had an analysis platform, and we wanted to optimize dosing for subsequent cohorts. So the way we did this was we developed an automated R platform for trial simulation and model evaluation. We then, for the trials, we simulated all our subjects into a population, and from that population, we formed 10 different reference trials. From those reference trials, we took subsets of those trials for our designs, whether it was a 2-2-4, which again represents 2 patients at the low, 2 patients at the mid, and 4 patients at the high level of dosing. And we repeated this 10 times. And we compared these values back to the reference and back to the true simulation values. We did this by estimating the PK/PD parameters in non-min and using those parameters to estimate a % recrudescence over the dose response curve, which is this picture right here. We then fit this % recrudescence versus the dose response using a Hill function as described below, and then utilize PFIM for dose optimization, much like we would utilize it for selecting optimal sample times. We then simulated a second cohort of data with these optimized doses, combined them with the first cohort and re-estimated the PK/PD parameters, then re-evaluated the dose response curve. So these results correspond to our first poster that you will find linked on the site. And this shows that parameter estimates were comparable across the designs. Both the central tendency of these parameter estimates for the fixed effects were all relatively similar to one another when compared to the true value and compared to the reference value. The precision of the parameter estimates was also good for all fixed effects with percent SEMs less than 10%, which is what this plot down here is showing. So for the reference trial, you have the median and the 90% confidence interval, as well as for the 448 design and 224 design. And even though the 224 has the largest degree of parameter uncertainty between the confidence intervals, it's still less than the 10% as stated before. We were also able to simulate out from these parameter estimates the parasite growth with parameter uncertainty. These results correspond to our second poster, and the summary table here shows the PFIM selected doses. The ones in bold are ones that were in the first cohort, so they were automatically selected, and then we allowed PFIM to select 3 additional doses. The ones that are in red are the ones that PFIM selected most amongst all trials. So 600, 800, and 1,000 milligrams when the cohort 1 had 100, 250, and 500 milligrams. And this corresponds to the top picture, where you can see the red line represents cohort 1 and the blue lines represent cohort 2, and they're the median 90% confidence interval. So between cohort 1 and cohort 2 for the top, really the median value did not change significantly, but you do get some increased precision when adding the second cohort, as you can see in the confidence bands. The kind of takeaway picture from this is the second picture, where we chose doses that were not near the ED50, and so it had a hard time estimating that curve. And so if you had just done a traditional design, you would be stuck with the red line with no chance to correct. If you had just done a traditional design, you would be stuck with the red line, and you'd have no real chance to correct towards the true underlying percent recrudescence curve. But as we can see with the 224 and the 336 designs, having an additional cohort was able to shift the median values significantly towards the true simulation values. This is important because in these pictures, we can see that the amount of bias is dependent on initial dose selection, the number of cohorts, and the study designs as well. So for some conclusions, we were able to accurately identify dose response with a single cohort with 3 dose levels, which is different from the traditional design, which is 3 separate cohorts with those 3 dose levels. In addition, we were able, when we added a second cohort, we were able to increase parameter precision. And in the case where we were not near the ED50, we were able to get the median values much closer to the true simulated values of the underlying dose response curve. So now we have a scalable platform where we can rapidly and efficiently compare study designs and dose selections for subsequent cohorts in adaptive viz studies or phase 2 clinical trials for malaria. This platform is able to run anywhere on the low end of 10 trials all the way up to 100 to thousands of trials, and still have rapid and efficient comparison between these studies and designs. Thank you, and if you have any further questions, you can reach out to info@simulations-plus.com. Thank you for watching. You can find my poster and others like it in our resource center at www.simulations-plus.com. I look forward to answering any questions now.
Thanks, James. Great poster. We also have a special guest answering questions with James today, Dr. Joel Owen, Vice President of Pharmacometric Services at Cognigen. He has previously served as Director of PK/PD at Cognigen and was the Professor of Pharmaceutics at Union University in Jackson, Tennessee. So welcome, Joel, and thank you for joining us today. Okay, the question that came in, thank you, Carol, for sending this. You mentioned scalability and being able to perform tens to thousands of trials. James or Joel, can you explain that a bit more?
Yes, thank you for the question. So when we were developing this platform, we wanted to be able to test multiple designs. We proposed the two differing designs between the posters for this analysis, but we also wanted to be able to test a numerous variety of designs. So what we did was we made the platform so that we would be able to test against pretty much as many reference trials as we wanted, as well as many designs. So, you kind of compound on each other. And so if you have, 100 reference trials and you're selecting 10 designs from each of those, you quickly get into those thousands of ranges. But if there's particular designs that you are really curious about investigating, it scales down to that as well. So it gives a lot of flexibility and the ability to define what designs you're going to use, as well as how many stochastic replications of those designs as well.
Okay, and Shiva asks, do we have to calculate sample size in each cohort stratification for testing?
Go ahead, Joel.
So I think that through the simulations, sample size can be assessed. This is really an approach to make the maximum use of limited amount of data. So when we were thinking about the volunteer infection studies, they're really run as quite small studies currently in their traditional design, and a power calculation and sample size are not typically a part of that. If you want to explore the sample size through the simulation estimation approach that's in this platform, you could certainly do that and see what the impact is on parameter precision.
And Joel, was there any other key takeaway that we should take note of from today's poster?
Sure, I think the approach is a very valuable exploration of how alternate trial designs can be considered to make maximal use of limited amounts of data. So it fits really nicely in this volunteer infection study type of paradigm for studying malaria drugs. But I think for other PK/PD environment, we have an opportunity to think about novel trial designs and explore the efficiency of how we develop PK/PD response curves. And it could be applied more broadly, even beyond volunteer infection studies. I think we're at a place, particularly with all the COVID activities, the way the pharmaceutical industry is going to change, we need to be thinking strategically about how to make the most use of limited amounts of data. And simulation, estimation, modeling, these kinds of approaches bring great opportunity, great tools and strength to the table to change the paradigm of drug development. Particularly in volunteer infection studies and the way these trials have run, we have a real opportunity to decrease the length of the trials by at least removing a cohort, but also improving the amount of information we get from those trials. In the end, if we run a 2-cohort study, as we've discussed here, you would have up to 6 dose levels, informing the exposure response curve before you move into a phase 2 trial design. So it's an opportunity using these methodologies to make the most of the information we gain for informing future trial designs and understanding exposure response curves.
Thanks, Joel. I agree, absolutely. And an opportunity for collaboration with our listeners and users. Well, moving on, now we are going to go back to John DiBella for the highly anticipated GPX and APX software release due out later this year. Any questions related to the demo can be emailed to info@simulations-plus.com. And John, go ahead and take us home.
Thanks again, Cool Cat. So I'm just going to spend a couple of minutes going through a few slides illustrating what it is we have been focusing on over the past 12 to 18 months as we develop the next generation platforms for GastroPlus and ADMET Predictor. With GastroPlus version 10, GastroPlus X, GPX, there are really 4 themes: model flexibility, data management, software engineering, and user experience. We will always, always, always be stubborn about the science and continue to have a laser-like focus on providing you with the most accurate algorithms and underlying models possible. But we do appreciate that there are more and more scientists who feel comfortable customizing the models in a variety of different ways, essentially building fit-for-purpose type models. And so what we're doing with GPX is adding in some new model types for tissues and organs, extending beyond the options we have today for permeability limited or perfusion limited options. For those who are in either clinical pharmacology or pharmaceutical sciences, where your focus is typically on one specific area. In the case of pharmaceutical sciences, the formulation or absorption side. For clinical pharmacologists, the systemic exposure side. We will give you options to utilize minimal absorption or PBPK models so that you can do your mechanistic simulations on the area of interest. We will have flexibility to be able to dose into any tissue, and as mentioned earlier in this webinar, you'll also have the flexibility to be able to have multiple exposure routes simulated at the same time. All of this being done without the need to write any software code. In terms of data management, we are going to be implementing some audit trail tools that will make it easier to log certain events, communicate what has been done on a model to new users as that project is being passed along. There will be new settings files available where you'll be able to save custom user specs, whether there's a certain GUI design that you like to use, whether there are certain settings to the models that you wish to have applied each and every time you start on a new project. You'll be able to save a user spec settings file. And then everything is going to be wrapped up in easy to share XML project formats. We will still have an underlying database, but all of those support files that we have been generating over the years will be saved as part of the database with the settings file then wrapped up into an XML project format. We will be introducing file converters to make it easy to convert your legacy GastroPlus database and support files into this new format. And this should make it a lot easier to be able to share and upload projects to health authorities or regulatory agencies for more efficient review. From the software engineering perspective, there will be command line scripting options available in Windows and Linux, or at least Windows in the GPX first release. We've introduced multi-threading parallelization, so now we'll be able to zoom through optimizations, we'll be able to zoom through population simulations by leveraging all the cores on your machines. And we've also introduced some new optimization algorithms so that we can converge or deconvolute to final solutions much more rapidly. And then finally, with respect to the user experience, we've spent a lot of time having conversations with current users of GastroPlus, different levels of experience, different departments, different markets, using that feedback and working with a UX design consulting team to help us redesign the user experience. And so what you're going to see here on the next couple of slides are some screenshots of what that new user interface is looking like. We are going away from the traditional GUI design from the late 90s, early aughts, where you had a menu-based system going horizontally across the top of the program, that being replaced now with a left side navigation menu. We have spent a lot of time trying to more logically order the various inputs to the models in a way that just will make it easier for you to identify where you need to go in the program to make any changes. You'll see here that there are, for the home page of the interface, different colors. There's also options for you to customize fonts or font sizes as well. That would all then be saved with the specification files. If I zoom in a little bit, so here, for example, looking at the physiology window, each of the areas on the user interface will have accordions that separate the different sections, which you can collapse or expand and go in and review and modify all of the inputs on just a single view. Here we see the extension of the gastrointestinal model view, where we've got our, of course, 9-compartment ACAT model. We will have sections where we can modify some of the physiological parameters associated with a specific compartment. We also have the traditional grid-like view that we've all gotten used to over the decades. And if I move over and take a look at one of the main compound pages, you'll see here, for example, we've got our inputs categorized based upon different processes or mechanisms. And with, for example, here, the expansion of the precipitation section, we see all of our inputs that would be associated with precipitation in one area. There's no longer any need to go to a different menu item, open up a different window to modify anything that would be specific to precipitation. Same thing applies to solubility, permeability, and so on. And just quickly here, taking a look at the PBPK window. For those who have experience today, you set up a PBPK model, you have your summary grid on the main interface, but you then have to click on a button to go and modify the PBPK model itself. Now you can see here everything in one accordion. You have the model editor where you can change between different tissue model types, add tissues, delete tissues. I can review the tissue parameters on the right side here, and also leverage the summary table looking at KPs, clearances, and so on. And finally, from an output perspective, We have the ability to run various simulations, have on the main window the actual predictions of plasma concentration time profiles with the summary results here, and also be able to take a look at individual subject profiles along with population outcomes if we're doing population predictions. So we're really excited about the direction we're going with GPX. Hopefully it should be available in your hands in the very near future. I know that we're at the 9:30 mark. There was a really nice webinar that was hosted yesterday on HTPK simulations and ADMET Predictor. And as part of that webinar, we also discussed a little bit some of what is coming in APX. I think I would encourage you to review that webinar so we can try and end this today on time. Arlene?
Thanks, John, and thank you to all of our presenters and participants today. Although some of our face-to-face events might have been canceled or postponed, we have many virtual programming opportunities that you can take advantage of, ranging from free webinars like yesterday's HTPK webinar to virtual masterclasses, next week's 5 free GastroPlus and advanced workshops, courses to support your pharmaceutical research and drug development efforts. You can find registration links and today's webinar replay at www.simulations-plus.com. Have a great day, everyone, and stay safe. Thank you for joining.